This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 13.
Ooh.
Recorded on July 14th, 2010. Bastille Day, no?
Yes.
Hi, everybody. I'm Vincent Racaniello, and with me is Dixon Despommier.
Hello, everybody.
Welcome back, Dixon.
Thanks, Vince.
You've been away a long time.
I have.
And as a consequence, TWiP has not been published.
I feel badly about that.
Don't feel too badly.
I don't.
A little bit. A micron of badness.
I feel empty inside that I haven't fully participated in the dispersal of this knowledge. How's that?
Your fans await, as they say.
How flattering.
1 month, but here we are back with a TWiP on July 14th, Bastille Day, and the 13th episode. You know, on TWiP, we're up to 90.
I do know that.
It's impressive, isn't it?
It is.
You know what happens at 100?
I guess we have a party.
Everything explodes and it's destroyed.
That's right. We start again. Start renumbering.
So the last time we did TWiP,
Yep.
we introduced Toxoplasma gondii, which you say is one of the most successful parasites on the globe. You think it's the?
Among mammals, it's the most successful.
Amazing.
It is. Bar none. This is just absolutely a given.
Okay. You're the boss. I'll take your word for it. And today, what shall we do, Dixon?
Well, I would like to talk about the clinical ramifications of acquiring toxoplasmosis and also the pathogenesis, something about how this parasite actually succeeds and why, in its most benign form, is a silent partner in our lives and basically plays no role that I can think of whatsoever in terms of the way we carry out our daily lives. However, there are some nuance here. One of them involves cats and mice. And what happens to the mice?
Can we review? The cat serves what purpose in the life cycle?
The cat is the definitive host.
The definitive host.
Because it acquires the stages from its prey.
Sexual cycle.
Which is exactly right.
That's exactly the definitive host, where the sexual cycle occurs. And then the mice and the humans and whatever other animals are
they're
accidental.
Considered intermediate hosts.
All right. And here in the U.S., many cats are infected.
Many.
And your pet cat might be infected.
That's true.
It may or may not be ill.
Yes, and we discussed the fact that even though your cat doesn't go out and hunt, which cats will do if you let them, cats can still acquire toxoplasmosis inside the house.
Mice?
Not because you have mice. For some other habit that we ourselves are guilty of. Imagine yourself in the kitchen, and you're a cat lover, of course, so the cat has full access to the kitchen, and you're making hamburgers. How about hamburgers? Okay, raw meat. And it hasn't been frozen. That's the one caveat. If it's been frozen, there's no danger here, okay? But if it hasn't been frozen, if it's freshly ground and taken home from the butcher's, and you're sitting there at your sink and your countertop, and the cat, of course, jumps up onto the counter to see what you're doing because they're very curious animals, you're making hamburger patties, okay? And you would never, never, never in 1,000 years feed any of what you will eat to your cat. You won't do that. But in this little package of meat, there's always a little scrap or two left. And, of course, well, why don't we just feed that to the cat then? How cute. Oh, look, a little gift. And you put that little in the center of your hand, and the cat will lick your hand and be grateful and start purring and wanting more. This raw meat could contain the pseudocysts of Toxoplasma, and that's how cats can acquire that.
How did the cow acquire the pseudocysts?
Not necessarily a cow, but a cow will acquire the pseudocysts.
Well, that's where the beef is coming from.
Yeah, it could acquire it through the accidental ingestion of cat feces, in which the oocysts, the sexual stage products, are produced.
So how much of the hamburger meat in the U.S. has
It's low. It's relatively low.
All right. Is it still a reason why we should wash our hands after handling raw meat?
Always. Well, there's a couple of other reasons too, but the biggest reason is it's unsanitary. But the point is that if you take all your meat and freeze it first, then you have no worries whatsoever about almost any of these parasites. In fact, even bacterial parasites are killed.
Okay.
Right. But most people would say, oh, that alters the texture and the terroir. Forget the terroir, kids. You're going to avoid parasitism this way, right? But remember, we also discussed national habits, and steak tartare turns out to be a big one in places like France and throughout Europe. And steak tartare, I mean, large portions of raw meat are consumed by single individuals. And it turns out that beef is not necessarily the source of that steak. It could be from lamb. And sheep have huge numbers of percentages of sheep infected with this because of their close association with farm animals like dogs and cats, but not dogs, just cats in this case. It's enough to allow an entire herd of sheep to
Why not dogs?
Flock of sheep to become infected. Dogs are canids, not felids. Only felidae takes the sexual stages.
And the sexual stage comes out in the feces.
Well, the products of the sexual stage.
And so that does not happen in intermediate hosts.
This is all true.
All right.
That's how you define them. Okay, so what happens then after you, as an adult, eats a piece of raw meat that has the pseudocysts in it? Let's say lamb, which is always served raw almost in the middle, as a rack of lamb would be. We like that. Okay, that's the way lamb is usually served. You eat this lamb, you acquire the infection because the pseudocysts are digested, releasing the organisms in the gut tract. They then penetrate the columnar cells of your epithelium, and the next thing you know, they're being taken up by macrophages in the lamina propria because that's their job. Little do they realize, of course, when they take these organisms up, the organisms have now captured the macrophage.
So far, a lot of similarities to Trichinella, except...
Or salmonella.
Yes, but we never talked about salmonella.
We didn't.
Trichinella does similar things. It gets into the gut tract, but it penetrates and goes on its own. It is actively pursuing a location, whereas these guys are passively taken up by monocytes, macrophages.
Well, they're actively taken up by them. I mean, once they encounter them, their job is to, there's somebody foreign, bam, kill it.
And once it's inside those macrophages...
Something bad happens. The macrophage starts to develop indigestion.
It can't digest these cysts. What are they called at that point in the macrophage? Not pseudocysts anymore?
They're called tachyzoites. The bradyzoites live inside the pseudocysts.
In the Brady Bunch.
The Brady Bunch, that's right. And then once they're released, each one of them is now referred to as a tachyzoite.
Interesting little structure. We had a picture of it.
They're fascinating.
Last time.
And they contain all kinds of interesting organelles. Like
is that a tachyzoite?
That's a rosette of tachyzoites that we see
each of these guys,
depicted on our websites, and each one of those, and it's inside of a fibroblast in this case.
Yeah, nice.
So why aren't they digested?
Well, the tachyzoite must antagonize in some way. Either that or it bypasses the endocytic uptake that would normally lead to lysosomal fusion, like viruses do.
That's number 2 reason, and there's a third reason that are possible here at least.
It produces materials that neutralize the enzymes. And
that's number 3. Number 4.
All these are wrong. These are all wrong?
That's right.
Does it kill the macrophage?
Eventually, yes. But it does so because it uses up all the resources that the macrophage contains in terms of nutrients for the developing parasite. The interesting part of it is that the parasite creates a niche inside of the parasitophorous vacuole, which prevents the fusion of lysosomes with that vacuole. So it alters the membrane in some way to make it incompatible with membrane fusion.
So it cannot fuse with the lysosome?
That's correct. And once that has occurred, the lysosomes can sit there forever and not be used. Ironically, not ironically, let's rephrase that too. Interestingly, if you feed macrophages in culture 50% live tachyzoites and 50% heat-killed tachyzoites, so that equal numbers are taken up by each macrophage, The dead ones will be digested fully by the membrane fusion with the lysosomal action of the release of acidification of vessels, etc. But the live tachyzoites in the same macrophage will prohibit that from occurring. And it does so in a very strange and wonderful way that we don't fully understand. The point is that it now captures a cell that's a transport cell. It's like hitchhiking a ride on a freight train if you're a hobo. And it can go anywhere it wants now, because the macrophage's job is to wander. It's called a wandering cell. And it wanders away from the lamina propria into the body, out through the circulation, and it could deposit this infection any place in the body, and it does. Once the macrophages are killed, and you get a round of replication inside the macrophage. Sounds very much like a virus infection or some other intracellular parasite.
Legionella.
Legionella, salmonella. You have a lot of parasites that behave this way, even tuberculosis. So then...
Here we go in your picture. Infected macrophages distribute tachyzoites throughout the body in an asexual cycle.
This is all correct. And in that sense, if you can now substitute the word macrophage for red blood cell, you can see how this resembles malaria. We've talked about that already. And in fact, they're both apicomplexa. They're both related to each other evolutionarily. In fact, they even contain some of the same organelles. But they do different things to their cells. Okay? So sorting all of this out using isopycnic gradients of sucrose and density gradients and this sort of thing has allowed science to fully, and of course getting the full genome, that you can now go back and look at, see which part is from the micronemes and which part are from the microbodies and which part are from rogue trees, etc. All these are fancy names for intracellular particles, each set of which has a different role to play in the evolution of the infection. One of the consequences of this is that every other cell in your body is now exposed to this infection.
Can go anywhere, basically.
It can go any place. And the fact is, the reason why I'm calling this the world's greatest parasite is because there are no cells that are immune from attack from this parasite.
So the tachyzoites are released as they multiply asexually. They're released from the macrophage?
Yes.
And they can penetrate other cell types, not just macrophages?
They can go anywhere. They can infect every cell.
How do they get into other cells?
The same way they got into the macrophage.
They're endocytosed.
Yeah, but that's an interesting question because Ron Fayer, who's a world expert at this and has worked out a lot of the mechanisms for cell entry and exit too, by the way, using related organisms like Eimeria, which is another apicomplexa that infects chickens and causes huge diseases. We often used to refer to that as the Frank Perdue disease. And other companies like Merck and Pfizer, etc., have spent fortunes trying to come up with drugs that interfere with Eimeria and coccidia in general to prevent them from taking away from the productivity of the poultry industry, right? So by looking at the way these organisms gain entrance and exit from cells gives you a hint as to what you might be able to interfere with. And so far, I don't know any drugs that interfere with the entry into a cell by Toxoplasma. So Toxoplasma induces the entry event. It's not an active process on the part of the macrophage so much as it is an active process on the part of the parasite to induce a special event mitigated by the release of proteins from the micronemes, primarily, and the rope trees. So once that occurs, you can go into a bipolar neuron in your eye. You can go into a astrocyte. You can go into a liver cell. You can go into
So the brain is not off limits, huh?
Oh, not at all. And in fact, that's one of the big consequences of this infection. Once it gets into your brain, because it's an immunologically silent organ, the parasite has its way with your cells, so to speak. Okay.
So, Dixon, if 100 people ate pseudocysts, how many would have clinical illness?
Probably none.
Ah.
That's the other beauty of this parasite, you see.
Doesn't hurt you?
It's benign.
Even in the brain?
It says, look, Vince, all I want to do is infect your cells. I don't want to hurt you.
Even in the brain, there's no consequence?
None that we can actually write about in a textbook and say, when you're infected with toxo as an adult, here's what happens.
So have you seen people die after a long life and in their brain is evidence of toxo and they never had an issue?
That's correct. This is exactly right. So you go to a country like France where steak tartare is a religion almost, and champagne. And you encounter these two items very early on in life and you never give them up because they're so good, right? when the AIDS epidemic hit. The thing that keeps this parasite in check, by the way, is interleukin number 12.
So we do make an immune response.
Well, I wouldn't call interleukins an immune response, but it's an acquired inhibitor. All right? It's an acquired inhibitor.
IL-12, you said?
IL-12.
And what does IL-12 do to toxo?
We don't really know too much about it. We could type it out and see what we get on the internet right now. Maybe there's something more known about it than when I first looked at this data. But I do know that when you get HIV infection, it downregulates interleukin 12. And when that happens, it exacerbates an already Toxoplasma present infection. And that used to be the big problem with catching HIV infections. It didn't actually kill you. Your other infections killed
Correct.
you.
Well, that's in the end what you die of, opportunistic infections.
Correct. You die not from HIV, but with HIV. AIDS. Okay. So if you type out interleukin 12 and Toxoplasma division, it interferes with division. It's basically like an interferon for the Toxoplasma organism.
Listen to this, Dixon. Production of the cytokine IL-12 by phagocytosis drives the differentiation of interferon gamma-producing cells during toxo infection. So that's what IL-12 does, which is turned on by phagocytosing the toxo, and that leaves the production of gamma.
Right. Interesting.
But they say the role of IL-12 in regulating memory differentiation during toxoplasmosis is unclear.
That's right.
So this is actually a recent paper, 2010, PLOS Pathogens.
The authors?
Wilson, Grotenberg, Liu, Zhao, Frickel, Goebbels, Pleu, Hiddepleu.
Pleu.
Yeah, Hiddepleu from Harvard and Yap.
Okay. All right, so people are still working on the connections between what the parasite induces and what the host induces. And so, but ultimately it achieves a balance, you see. So if there's not a lot of damage in the tissues prior to the time of the induction of IL-12, the parasite stops its reproduction cycle and develops into a pseudocyst. And the tachyzoites transform into bradyzoites. And the bradyzoites just sit there. And they say, I've got all day, all night, all day, all night, all day, all night. It depends on how long this host lives. But you know what I really want? I want this host to go from where they are now to someplace where I've never been before. And then you can die. You can live a long life. You can live long and prosper.
So they hang out as pseudocysts and bradycysts.
The bradyzoites are inside the pseudocyst.
That's right. There are many bradyzoites.
Many.
And this is sort of like a spore, right? It doesn't do much. It just
or it's like a
out.
Pseudotubercle.
Exactly. And it's happy to stay this way.
In this case, a tubercle with bacteria inside that don't divide because your immune system has got them in check.
How many of these pseudocysts would be present in an infected animal? Many, or just a few?
In your brain, you could have millions.
Wow. Millions of pseudocysts.
Well, it depends on how fast you make IL-12, right? It's all a kinetic war between how fast I can divide and reproduce versus how fast I induce my inhibitors.
And if I didn't make IL-12, if I had some kind of defect, what would happen? Then I'd be dead.
You died of an encephalitis.
So there are some people who die, right?
Yes, the AIDS patients, you see, because when you develop HIV/AIDS, it suppresses IL-12 formation, and out comes this organism from the bradyzoites, transform back into tachyzoites, and they kill you with an encephalitis.
What about transplant recipients who are immunosuppressed?
Yeah, that's true too.
They also
not only that, you have to watch out for the organisms in the transplanted tissue.
They could have toxo.
But of course.
They don't look for them when we transplant an organ, do we?
Of course not, because you'll find them. then they'll reject the organ.
Then you would never transplant anything, huh? How about a transplanted heart? Could be.
Tell me about it. It can be in any tissue, Vince.
So
any tissue.
These people are seriously immunosuppressed.
I know. I know. But maybe not from the standpoint of IL-12.
No, probably not.
Right. But HIV is specific inhibitors.
Yeah.
All right?
Sure.
Okay, so you've got some parasite games going on here, right? It's a war between two different parasites now. I mean, toxo doesn't want to kill you. HIV doesn't want to kill you. And ultimately, they both end up killing you. Because you put them together and you've got something more. You've got synergy.
Sure.
So it's a negative synergy. So anyway, let's get back to toxo, though, because if you're a normal adult with a normal immune system, which is highly activated and goodly, that's a bad word, and it's properly nutritioned, then the likelihood is that you will not suffer anything from an exposure to toxo. There are some exceptions here, of course, and I can tell you about some of them, and it depends on how many of the organisms you ingest to begin with. You can overwhelm a normal immune system, and so I'll give you the case history that's the one that we use a lot in our teaching, or used to use a lot in our teaching, and it involved 4 Cornell medical students who were really hot to get involved in, you mentioned heart transplants. Christian Barnard was the first surgeon that transplanted a heart. And the guy's name was Wyszynski. I never forgot his name, because he lived for like 10 days and then he died, right? But
I do remember.
And it was in South Africa.
It was, yeah.
So Christian Barnard used to go around the world giving lectures on how he kept alive a man for 10 days. Beyond his limit of staying alive because he would have died otherwise, right? So
this was with a big external mechanical.
No, it was somebody else's heart.
It was someone else's heart.
Yeah, he didn't have mechanical hearts in those days.
This was an actual transplant.
This was an actual transplant.
Yes.
So the point is that here are these 4 medical students. Let's make them second-year medical students. And there's this big lecture by Christian Barnard, right? So, but they have a lunch break. They've got to go to lunch, but they want to go to this lecture. They have 20 minutes to eat. Now, there's this little restaurant that used to be around Cornell called the Recovery Room.
I remember.
You do? Okay, good. Because, yeah, I mean, you used to go to Rockefeller a lot and Sloan Kettering, and I was at Rockefeller for 3 years, and I knew about this little place.
Did you go?
I never went to that little recovery room. No, I had some other good options. The entire East Side restaurant industry, right? So these 4 students, they rush off to lunch, and they order hamburgers. And they say, we need them quick because we got to go to a lecture. The guy says, okay, and he puts them on the grill, and he turns them and makes the hamburger out of it and gives it to all of them. And they ate it, and the meat was seared on the outside and raw as hell on the inside and not frozen. Well, that's bad, because about 3 weeks later, each one of them began to notice lumps under their armpits. That's a bad thing. Because now they heard the lecture by Christian Barnard. It has nothing to do with anything that would give you a lump under your arm, would it? But there was another lecture, you see, they heard about, which was Hodgkin's disease. And that gives you a lump under your arm. Many, in fact, and lumps on your neck. And in other words, you get lymphadenitis. And so each one separately came to the student health clinic to be examined for the presence or absence of Hodgkin's disease. Because that's what they knew they had. They knew they were going to die, and every one of them was just depressed as hell. And the same story came out of this. And that is, they took serum, they did a complete, as good as they could do in those days, examination of their blood, and they said, you don't have Hodgkin's disease. And you don't even have mononucleosis, which could be the other cause of this. You have Toxoplasmosis.
I actually looked.
Yeah.
What kind of lab test do you do for
an antibody? They do an antibody test.
Why would they look for toxo?
Because it's part of the differential diagnosis.
Did they know that there was a cluster of 4 students?
Of course not. That's how they found them. They said, wait a minute, somebody else came in with that same thing. And wait a minute, somebody else, and somebody put 2 and 2 together and got 5. Went back and interviewed all of them. They all ate together at the recovery room, and then they went off to hear the Christian Barnard lecture. So that's the basis for studying the evolution of antibodies and cellular responses to Toxoplasma. They used these 4 students as adult-acquired Toxoplasmosis.
And why were there lymph nodes under their armpit? What would those be called? Axial?
Yes.
Lymph nodes, why were they so swollen?
Well, because this parasite can infect all tissues, and since they must have received a whopping dose of bradyzoites in the form of pseudocysts, it overwhelmed their normal immune system. It went every place. And the immune system overreacted, of course, by producing more monocytes and macrophages and all kinds of other things.
What happened to these gentlemen?
Oh, they lived normal lives.
They recovered.
Oh, of course they did. But meanwhile, every week they contributed blood and cells for the laboratory to work out all of the nuance with regards to adult-acquired Toxoplasma. And they saw IgM antibodies come up and go down,
Yeah.
and then they saw IgG antibodies come up and stay up. In fact, they never went down. IgG levels remained high throughout their entire infection period.
Because the parasites are there?
Because the parasites are there, yes, and they occasionally come out of their cells because your immune system does wax and wane, no question about it. A little bit comes out, bam, stimulates the immune system, back up goes the IgG levels, but the IgM levels go down and stay down. So they learned from these 4 students, and from other test cases as well, but mainly from these 4, that if you acquire toxoplasmosis on day 1, by let's make it day 20, you've got detectable levels of IgM antibodies. That lasts for about 20 to 30 days. In the meantime, you start to detect IgG antibodies, which come up and replace the IgM response, and it stays up for the length of the life of the person.
What was the period of What was the incubation period from eating to the swollen lymph nodes?
About 2.5 weeks.
Okay.
About 2.5 weeks. So they call that form adult acquired.
Right.
And usually there's no sequelae, no nothing. All right.
And that can happen today.
It could. It still is happening today. Of course it is. There's no vaccine for this. So, yeah, that's it.
And how about the recovery room? What happened to them?
Well, they were told either to freeze the meat or to cook them longer, one of those 2 things, because that's where it was caught. I think that's since closed for other reasons, probably rodents or something like that. Maybe that's where the toxo came. No, that's another subject.
It could, right?
It could. It's unlikely, though, because they don't do their own slaughtering of animals in situ. So the point is that that there's a good immune response.
Still there, Dixon.
It is still
there. The Recovery Room Pub is on 417 East 70th Street.
I bet you it's under new ownership.
In New York City.
You know, they're going to start grading restaurants in New York, Vince. Have you heard that?
I'm sorry, they're going to start what?
Grading restaurants. The health department is going to start grading
restaurants.
Really? I'm not serious, but they are. There are 24,000 plus restaurants in New York City, and they're going to get a grade from A to D. And the highest grade is a good, and the lowest grade is terrible. You know, just like in school.
That's funny.
And they're going to post the grades in the windows.
Here's a picture of it. It's right next to Murphy's Law.
That figures.
It's a good name for a pub, right?
I'll say, especially if you go in there and ask them for some toxoplasma. Okay, so back to our story.
That is adult acquired toxo, which is right here in your book.
It's the most commonly seen form of the disease. All right, it's a disease, sure. The lymph nodes swell up, but then eventually they do go down. And when they do, they stay down.
Okay.
They also induce a very funny-looking cell, which is reminiscent of cancer, but it's not. So you can't tell by just doing a biopsy what it is. You can tell what it's not, but you can't tell what it is. You have to do some more tests. They used to be able to take a mouse raised in pathogen-free conditions, immunosuppress it with cortisone, and take a little biopsy from the lymph node and then make a homogenate out of it and inject it intraperitoneally into the mouse. And if it's toxoplasma, the mouse will become inundated with toxoplasma. In another week, you can isolate them and look at them using indirect fluorescent antibody stains. In all the cells, you can find them, okay? And that used to be one of the tests. The IFA test was. But now we have lots of different things like PCRs and, complement-fixing antibody tests, and we have a lot of good options for telling indirectly whether the parasite was ever there or not. That's an antibody test. If you want to know whether it's there or not, then the PCR test is what they use now to tell. But it costs a little bit of money. Some hospitals don't have the right facilities. The option is, of course, to distinguish it between Hodgkin's disease and mononucleosis. If you can say it's not either one of those 2 things, the chances are that it's toxo.
And then you go home and that's it.
Well, they give you some drugs. They'll give you a pyrimethamine or something like this.
What's that?
It's a drug that interferes with its PABA synthesis.
Oh, so there is a specific toxo drug.
Oh, sure. Oh, yeah, lots of them. You wouldn't dare give that to a woman who's pregnant, however, and she may very well turn positive during her pregnancy. If that's the case, then we can talk about the second form of
Why would she turn positive?
Because she ate meat that contained the...
I see.
She started out negative. In other words, if you become pregnant, all right, Now, that excludes you and I, but not our wives, okay? In the early days of our careers, if our wives became pregnant, then what they should have done is been tested for the presence or absence of Toxoplasma antibodies. Now, if they were present, you don't have any worries at all. She can eat all the raw meat she wants during her pregnancy, and none of those parasites will ever cross the placental barrier because immunity in the mother protects the fetus.
Right.
And we don't fully understand that mechanism either, but it does. And there's huge amounts of evidence for this. It was done mostly in France by Despommier back in the 70s. If, on the other hand, she's negative for Toxoplasma, and in many places, like, say, France, for instance, you'll never find a pregnant woman that would be negative for Toxo because of the relatively high level of infection in that country. Maybe less than 10% of the incoming pregnant women to a clinic would test negative for Toxo, and the rest would all test positive because they've encountered it prior to the time they either matured or got married or became pregnant. In the United States, it's about 20 to 25% of the teenage and early 20s female population which would test negative.
Seronegative.
The rest,
Or positive?
60 to 80% test positive.
So you and I are probably positive?
Probably.
Wow.
Probably. You like your steak medium rare, don't you?
Yeah,
me
sure.
too.
I've had red centers, yeah.
Oh, but of course.
So you and
And
I
you've-
probably have
We should get ourselves tested.
pseudocysts in us?
We probably do.
All right, that's it. I'm done.
Now, there's a guy, wait, I'm going to tell you something. Well, I'm spoiling a lot of stories here by jumping ahead, but a guy by the name of Jack Remington, who is a fabulous researcher at Stanford. I hope he's still actively doing research. We could type his name out and find out. I think he is.
What's the name?
Jack Remington.
Jack Remington.
Yeah, he's produced a lot of literature on Toxoplasma. He's an amazing investigator. I remember when I was on study section, his grants were always couched, I produced X number of papers per $10,000 of grant money.
His last paper was in 2002, a test for Toxo IgG for confirmatory testing of pregnant women.
Right. That was his big push. That was what he really specialized in. And he may be now retired, just like I am, but he was a world leader in Toxoplasma.
So why do we worry about
I'll
whether
tell you,
women are...
Vince, it's not because of the mother, it's because of the fetus. If the mother doesn't have protective antibodies for the fetus, then if she becomes infected during her pregnancy, it depends on when during her pregnancy she becomes infected as to what happens to the fetus. Just like rubella. So this is a virus story. Okay, this could go on TWiV as well as TWiP. This is a TWiV-TWiP combination. It's a hybrid.
It's a crossover.
It's a hybrid. And the story is the same. It's exactly the same. If the
rubella,
fetus,
it's the first trimester.
It's the same with Toxo too. But it's more likely to happen with rubella than it is with Toxo. Tell me why, Vince.
It's probably easier for rubella virus to get across the placenta than
Well,
Toxo.
tell me about rubella virus. How many viral particles are produced during a rubella infection?
Millions and millions and millions.
There you go. And they're all floating free in the serum.
Free in the blood. Very small.
Right.
Easy to get into the fetus.
Right.
Infect and cause.
But do they get across by themselves, or does
defects.
something take it across?
Not clear, Dixon.
But
it could go over in a cell. It could go across. Capillaries are very small.
Can rubella?
By definition, of
true,
course.
but those are not typical for the placenta. There's something else present there. But let me ask you this: Does rubella cross the blood-brain barrier in the brain?
Probably.
I don't know that for sure. But
I'm going to look it up right now.
If it does, then it's probably taken up by something and pushed across rather than get across by itself. So these barriers that are set up, like the kidney, for instance, with regards to circulation, or the choroid plexus, or the placenta, they all have restrictions as to what can go across, right? In fact, some drugs are so big that they don't get across because they don't diffuse through these loose junctions for the... Actually, the capillary beds are tight junctions, not loose junctions.
The form of Toxo would be the tachyzoite.
Correct. Inside of a cell that then diapedeses through from the mother to the child.
Yeah, squeezing through. Diapedesis.
That's right. Well, actually, right through. Doesn't squeeze through. It goes right through the cell.
Well, diapedesis isn't squeezing through the junctions. In the brain, it is.
Well, maybe, but I've seen movies of this.
That's what I've been teaching medical students for the last 20 years.
Well, but the cell can actually go right through another cell by the same process that toxo gets into a cell and goes out.
So it's always within a cell that it goes over the placenta then.
Correct. And the reason why we know this is because all of the markers for those macrophages are present before they diabetes, but they're absent when they come out the other side, Vince.
Mm-hmm.
They lose them.
Why do they want to go in anyway? What's the purpose?
To go through and get to the other. It's like the chicken. Why does the chicken cross
I presume the macrophages are patrolling and they're looking into the fetus.
It's their job.
Okay. And if they have these tachyzoids in them, they bring them into the fetus and then
Bingo.
Problems.
Now, so if you're in the first trimester, second trimester, or third trimester, it matters as to the consequences to the fetus.
Mm-hmm.
All right. The most damage you can do to a fetus is during its early developmental stages, right?
Right.
So the first trimester is the one everybody worries about. But the placental mass is only so small that the chances are low that the mother will transmit her infection to the fetus before she develops protective antibodies.
All right.
So the number of fetuses that become infected during the first trimester are low, but the ones that do become infected are severely affected.
Mm.
They develop chorioretinitis in their eyes. They develop plaquing in their brains, as you can see. I have a picture of that here. They develop hydrocephaly.
Mm-hmm.
Now, hydrocephaly is huge. Huge. That's not a pun, kids. That's just a sad deal. Janku and associates in the then Czechoslovakia saw the first cases of Toxoplasma that created hydrocephaly. And everyone thought it was such a rare disease. And in fact, there was a guy at this pathology department. His name was Wolf. And Wolf actually proved that hydrocephaly, at least some forms of it, were caused by Toxoplasma infections. So it's one of the consequences of early infection with Toxoplasma as a fetus.
Here it says in your table, after congenital,
Yes.
so congenital toxo after maternal infection.
Right.
First and second trimester, 73% will be fine. They won't get infected.
Right.
The fetuses. 13% subclinical, meaning no symptoms. 7% mild infection and 6% severe.
That's right.
Infection.
And it's only because the placental mass is so small that the macrophages, with the Trojan horses, so to speak, don't encounter the placental mass before the maternal antibodies are induced. The older the fetus, the more likely it is, because the placental mass is now large, that the macrophages should become infected during that time.
Mm-hmm.
They will migrate through and give the fetus the infection. But the fetus is now beyond certain stages of development and is less and less affected by the outcome of the infection. However, even during the latter stages of the third trimester, and if the fetus becomes infected with toxo, there are some very subtle changes that the parasite induces, which are more or less permanent and related to cognitive recognition and visual recognitions. And so, but I actually participated in a grand rounds here once where Dr. Gold, who's one of our world famous child neurologists, presented a case of a congenital toxoplasma in a 12-year-old girl whose eyesight was severely affected. She had to wear eyeglasses that, as they would say, are as thick as Coke bottle bottoms, right? And he said, I would like you to see what this little girl can do now. She's 12 years old, and she was born with severe congenital toxoplasmosis. So severe that the ventricles in the brain were enormous. Now, they're usually small. Okay, that's where the cerebrospinal fluid is produced. These were huge because of the erosion of the tissue by the parasite before IL-12 took over and stopped it. You'd say that with that much brain tissue missing, this person couldn't possibly be normal. And he said to her, before she spoke or anything else, he said, let's call her Melissa. Melissa, would you please go to the blackboard and write out the quadratic equation for me? And everybody else in the audience sat there and said, oh my God, I hope he doesn't ask me to do that because I've clearly forgotten it. And she walks right up to the blackboard and draws out, A plus B equals A squared plus B squared equals whatever the quadratic equation is. And Vince is going to type it out right now for us. So we can all know what the quadratic equation is.
AX squared plus BX plus C equals 0.
Thank you. And that's exactly what she wrote on the board.
Wow.
And she turned around and she said, will there be anything else, Dr. Gold? In perfect English, of course. And she was totally intact except for her eyes.
Mm-hmm.
Her brain had completely restructured itself in terms of its ability to store and process knowledge, which should tell us something about neurology in general, and that is that we don't know. Should I use that word, Vince? We don't know about the brain. We don't know anything about how the brain functions, although we fool ourselves into thinking we do. This organism affects the brain severely in some cases, and here's a perfectly normal, on the outside, person. You take a CAT scan of their brain and you see these enormous ventricles. She's missing a lot of brain tissue, but apparently it didn't matter. The brain found another way to do the same thing. And there's a lot of backup systems that we are totally unaware of. These organisms force us to rethink the way we think, even. How's that?
Mm-hmm.
Now, okay, so, but a lot of cases do suffer greatly, and some people actually die. A lot of stillbirths are caused by severe Toxoplasma infections. And severely damaged individuals are certainly produced throughout the world.
Sounds like it's more severe in these cases than in adults, right?
This is exactly right. But there are some exceptions to even that, of course. With a severely immunosuppressed person, then Toxoplasma will kill you.
Sure.
So let's talk about that for a moment, because that's a reactivation of
That's
Toxoplasma.
Called recrudescent toxoplasmosis?
Not recrudescent, because we know where it is. We know what caused it. In this, recrudescence refers to a malarial species, right? Plasmodium malariae, and we don't understand why it relapses, so we call it recrudescence. But we know why toxo relapsed in these patients, because they all acquired AIDS, the HIV AIDS virus. And as the result, it downregulated IL-12, which released the bradyzoites from their division inhibition. They transformed into tachyzoites and burst out of the pseudocyst and started to replicate. And about 30% of all the AIDS patients in France at one time were dying from reactivation of their Toxoplasma infections that they acquired as kids. Right? So you're a kid, you take their tar with the rest of the family, and you think you're normal, then you go off and acquire HIV, and the next thing you know, you're dead. So once they understood this, then they gave them a compound called Bactrim. Bactrim. It's a combination drug. It's a sulfur drug, and it's a pyrimethamine drug. So the pyrimethamine works against the PABA cycle, right?
Of the parasite.
Yes. And then we have a salvage pathway for that, so we're okay.
PABA meaning?
Para-aminobenzoic acid. And the other cycle that we interfere with from the sulfur drugs, of course, is the same cycle from another pathway. So you've got two ways of interfering with it. And so what we want to do is then, once a patient was diagnosed with HIV in France, they just started to treat them with Bactrim. And the rate of encephalitis went down to 0.
Of course, they still have AIDS, so...
That's correct, but interestingly enough, they could have actually cured these patients of their toxo infections this way. So they had none left at the end, right? So
is that right? It would cure them?
Yeah. Yeah, because all the parasites become activated and start dividing, and these drugs act against dividing forms. If you give it to a patient that doesn't have dividing forms, none of these drugs work because they're not synthesizing anything.
Right. Makes perfect sense.
Right. So we think we have explanations for all of this, but maybe there's some more to be learned.
So toxo is globally distributed.
It is. It's everywhere.
Are there no foci in any particular countries?
You mean, are there island communities that have none?
Or Africa has more than anybody else?
France.
France, because they like raw meat.
Yeah, and
many countries, poor countries, don't eat meat, so they don't have it, right?
Well, I wouldn't say that. They hunt. A lot of bush meat.
Well, if there's no
wild
place
animals
to hunt in the desert, then you can't eat.
Well, that's true. And so therefore, wherever you have populations that have vegetarian habits, I mean, you think about India, for instance, you'd think there's no Toxoplasma in India because nobody eats meat there, right? Well, they do eat meat, but let's say in the southern half of India, it's largely vegetarian, but they do keep pets. They keep all their animals alive.
But how do you get it from a pet?
Cats.
From the feces?
Yep. It contaminates your vegetables, and next thing you know, you've got Toxoplasmosis.
So are there island communities without toxo?
We've looked for them and haven't found any, but maybe we should type this out on the internet now and see what we get.
Dixon, I'd be happy to charter a boat and go around the world and look for such communities.
Yeah, I think Craig Venter would probably want to do that one before you. But
Craig.
So if you say Toxoplasma-free human populations, I bet you don't find any. I don't think there are any. Remember what we talked about with Trichinella? We talked about Eskimos briefly because they eat raw meat, and their name means raw meat eater. And there's a lot of Eskimo populations that have large amounts of Toxoplasma in their
Eskimo,
tissues. Yeah, sure.
What does Eskimo mean, Dixon?
It means raw meat eater in their own language, Vince. I just said that. I just said that.
I thought you said it 10 episodes ago.
Yeah, no, I said it just now, too.
Okay.
It's okay.
I was typing something.
Yeah, you were distracted. I understand that.
There's nothing I found about free populations that would
there aren't any. I don't think there are any.
But here's an interesting statistic.
Yes.
10% of U.S. people 6 to 49 years of age and 11% of women are seropositive.
Right.
IgG.
Right.
Wow.
Right.
It's estimated that between 30 and 65% of all people globally are infected.
Now that's a big number, Vince.
But there is a large variation between countries. In France, over 88%
right.
is infected.
Right.
Germany, Netherlands, Brazil also high.
Yep.
22% in Britain, and South Korea 4%.
4%
Yeah.
in South Korea.
Why is that?
It might be because they cook their meat well or they freeze it.
And no cats.
Oh, they have a lot of cats, but they eat them.
Are you serious?
Yeah, dead serious.
Wow.
Cats are eaten all over the world. They're a food source. Dogs too, by the way.
Sea otters have toxo.
Yeah, it's actually a big problem in sea otters. We talked about that briefly when I talked about the introduction to this
Dolphins,
subject.
whales.
Yep, cetaceans can acquire it too.
Anchovies. They're little fish, Dixon.
I know what anchovies are.
They travel from estuaries into the open ocean and probably spread the parasite that way.
Because the pseudocysts, not by pseudocysts in this case, by the oocysts, which are environmentally resistant stages. They passively transfer it.
Can you name some notable people who had toxo?
No, but I bet you can, because you've got the list in front of you and I don't.
You don't know any.
Well, regale us.
Arthur Ashe.
Really?
He developed AIDS, of course.
Yeah.
And he had toxo.
Yeah.
He had neurological problems.
Yeah.
In
fact, he died.
Sebastian Coe, who was a middle distance runner from the UK. I remember him.
Yep.
Martina Navratilova.
Oh, yes.
Retired from a competition in 1982 with symptoms of a mystery virus that was later found to be due to toxoplasmosis. So she probably ate raw meat, right?
During the French Open. I just made that part up, but it sounds like
I don't know where she was. Louis Wayne, an artist who was famous for painting cats. He later developed schizophrenia, which some believe was due to toxo resulting from his prolonged exposure to said animals that he was painting.
I'll be darned. Now, remember, I don't know if I mentioned this or not, but I'll say it again if I didn't. No, I'll say it again if I did. I won't say it again if I didn't. I'll say it for the first time. The effects of Toxoplasma on other animal species is different than it is on us.
How so?
Well, I'll describe an experiment that led to this conclusion, and that is that if you infect mice with Toxoplasma, which is easy, just take some cat feces with the oocysts and sort of put it in the mouse food and let them eat, they'll become enormously infected with this thing, okay? Now you've got two cages of mice. You've got one that's not infected, one that is infected, and you've got another large cage with a cat in it. Take the mouse from cage one that's not infected and put them in the cage with the mouse, and the mouse will instantly run away from the cat. Instantly, because it knows what's going to happen. But if you take the mouse that's infected with Toxoplasma and put him in the cage with the mouse, it actually runs towards the cat. What is that?
Runs towards the cat. What does the cat do?
The cat eats the mouse, of course.
It gets toxo.
It loses its ability to smell the presence of a cat. Toxoplasma actually interferes with a rodent's. This is true for rats too.
Mm-hmm.
It loses its fear of cats, and it does so by interfering with the ability to smell the cat.
It's all based on smell.
Yeah. That's remarkable. I think Richard Axel here should know about this because it's a remarkable study, a remarkable set of studies. So it's a parasite-induced behavior that favors the spread of this parasite to its definitive host. Now, we discussed one other like that, didn't we? When we discussed the worm that induces fat frogs. Remember when we talked about frogs? And
yes.
frogs' legs and parasite tails. I just sent you an updated version of that, by the way, a pictorial updated version. So it makes a frog fat so it can't run as fast, so the cat catches it, so it catches the tapeworm. In this case, it takes away the fear of the cat from the rodent, so the rodent is... Sure. It's a remarkable series of biological interconnectednesses that thrills and excites.
Dixon, so this happens in laboratory mice.
Yeah.
What about wild mice out there?
I guess
it might not. It might not.
It might not. It's true, it might not. But the problem with saying it might not is that you're not following Occam's razor very closely. The more likelihood of this is that it does happen.
Tell us Occam's razor.
Well, if you're faced with a choice between a simple explanation and a complicated explanation, take the simple one until otherwise proven guilty. Because now, there are complicated explanations of things, of course, but in this case...
But, it's an attractive idea for how the parasite is spreading.
So how would you go about proving this? What would be your laboratory-based field study? Oy.
It's a tough one, because all the infected mice get eaten, Dixon.
That's not true. That isn't true, but you could set up a series of experiments in which you trapped field mice. Trap them, and do a test for toxo, and separate the toxo from the non-toxo mice, and mark them with something that you can then recover them again from. And you can study the rate of predation between marked field mice that have toxo and marked field mice that don't have toxo. Just recover them.
This is a good experiment. This illustrates why controls are important.
No question.
You have to mark both mice, because if you just mark one, the cats are going to go up.
That's right. And you have to use the same color marker on the same place on the mouse for both the uninfected and the...
sure.
You have to have a way of distinguishing them, of course you do.
I mean, that's an experiment where you're actively participating because you're capturing, tagging, and releasing. And some would argue that you're interfering by nature by doing that.
But of course you are. So what's another way to do it?
An observational experiment where we don't modify the mice in any way.
We don't have a remote sensing device that we could point at a mouse and say, aha, this one's infected, aha, that one's not. And then we don't have another device that would make a little... I don't know. If you make a mark on an animal, you're altering its behavior, probably.
The only thing, and the thing also with your experiment, it's a good experiment, but you're not actually seeing the infected mice be eaten, so other things could happen to them, because you're just looking at a numbers game.
You have a third group.
Which is?
Which is a group that doesn't have any predation associated with it whatsoever. They're fenced in, but in a large area. So you can measure the death rate of these animals independent of predation.
Okay, that's fine.
All right.
But what if a hawk decides to eat the infected ones? There are many complications.
We have a lot of listeners.
That's why I like the lab.
We've got a lot of listeners out there.
Yes.
And we've got, I'm sure some of them do field work, and some of them have thought about how you would translate laboratory results into the field, into the real situations, right? Mm-hmm. Design this experiment for us. Come on, help us out here.
All right, we give the winner a TWiP t-shirt. How about that?
Ow! I would do better than that. I would go and give them a West Nile virus book or something like that.
Okay.
Perfect.
Do you have some West Nile books?
I do. Happen to have a few left.
All right. So if you make design an experiment to test the hypothesis that in the wild mice infected with toxo are more likely to be eaten by cats, wild cats, I presume.
Feral cats.
Feral cats.
Yeah.
And design an experiment, and we'll look at them, and the best one will get a West Nile book from Dixon, autographed.
Right. Maybe there's something that you could tag the Toxoplasmosis infected mice with that would accumulate in the feral cats, and you can measure the accumulation of this marker.
You can, sure. That would be good also. I think it could be argued that it would interfere with the whole thing. Schrodinger said, by observing, you're changing the whole equation.
Heisenberg says you can't do it anyway.
Heisenberg, not Schrodinger. Sorry, Heisenberg. Right.
That's right. Schrodinger had something to do with a cat, though. You're right.
Now, if we get no entries, it's because no one wants your book.
Or nobody's listening to this broadcast.
Which is possible.
I think it is. But
I enjoy talking to you, Dixon.
Vince, we would do this anyway, wouldn't we?
It doesn't it's not much extra work to record it.
No, but there's missing 2 items: a sandwich and a glass of beer.
Well, we could take care of that, Dixon.
We could.
If you want to record over lunch, maybe it would entice you to record on a more regular basis.
No, I'm doing it as much as I can now.
I'm sorry, I'm not trying to pick on you.
That's okay. So, but I'm interested to see what we get from our audience because like Car Talk, which is my favorite radio show, by the way, we could imitate the mantra of Car Talk because they always offer a puzzle at the end of their show. It has probably nothing to do with the show itself, but it's just a brain teaser.
And what do they offer in return?
Nothing. They offer, like a recording of their last 12 shows or something, and they said it's totally worthless, by the way, and you'll never want it, but should you just decide to enter this contest...
They have a lot of listeners.
They have a huge number of listeners.
What's their name? Click and Clack?
Click and Clack, yeah. The Car Talk guys. They're both MIT physicists. That's an amazing group.
We're Columbia professors.
We are. Come on, we can match them.
Why can't we get Why can't we get a radio show?
Why can't we get on their show? If they have a lot of listeners, let's just go on their show. Come on, Vince.
They don't need us, Dixon.
No, well, maybe cars and Toxo are related to some... No, that's not true.
I have to listen again. I listened years ago.
They're fun. They're a lot of fun.
So we did adult acquired, we did congenital, and this reactivation.
So those are the 3. That? Yeah, we just did, because if you acquire, if you're immunosuppressed for whatever reasons, and HIV is one of those reasons, but there are others too, right? Organ transplant donors and organ transplant recipients, they're linked in some way.
So it seems to me, Dixon, that you should check a donor for antibodies.
You can do better than that. You can do PCR.
But Dixon, since 80% of some populations are positive, you would never have a organ donation program if you threw out the ones.
No, you wouldn't. But what is the answer to that problem, Vince?
Don't eat raw meat.
No. Bactrim.
All right.
Treat them with Bactrim after they get the organ. Because they're going to get an infection.
Do you know if this is done? Yeah.
I think it is done prophylactically now.
All right.
There's another infection that acquires like this, and it's called pneumocystis. It's not a protozoan, it's a fungal infection, but pneumocystis is also treated with Bactrim, and pneumocystis was one of those other infections that AIDS patients would acquire. They didn't acquire it, they already had it, but it would come up in their lung tissue, and it's called interstitial pneumonitis. And Bactrim was used, by the way, that's probably how they discovered that it also could be useful against Toxoplasma.
Okay.
Because it was an original drug used in this fungal infection.
Dixon, is there anything else we need to cover here?
Well, we didn't cover the pathogenesis of this organism. That is, how does it accomplish this remarkable feat of remaining silent for these many years without being digested by the host? What is the mechanism at work which prevents the fusion of lysosomes to the lysosomal vacuum?
So that's the basic question. In every cell where these organisms are,
that's right.
The lysosome is prevented from fusion with the vacuoles containing...
That's right.
What's the name again, Dixon? The tachyzoites are inside the vacuoles?
Correct. Now,
do we know the mechanism?
We know something about the mechanism. Now, there's a guy by the name of John Boothroyd.
Yes.
At Stanford. And he's a wonderful, he was an associate, by the way, of Jack Remington's, and has done a lot of seminal work on the molecular biology of Toxoplasma, and has discovered in his studies that Toxoplasma actually secrete proteins which enter the host nucleus.
And what do they do there, Dixon?
That's a very good question. Well, you'd have to ask John that question, actually. But I do know that we made a connection between our 2 research groups when we found out that not only does Trichinella do this, but Toxoplasma does it too, and they're totally unrelated organisms.
Hey, listen to his most recent paper, Dixon. Toxoplasma protein number 16 subverts host function by direct tyrosine phosphorylation of STAT6.
Ow!
So it's modifying host. Now, STAT6 is a transcription protein that is involved in immune responses, early interferon type responses.
Right.
So it is interfering.
So wait a minute. We've now crossed the barrier between viruses and parasites.
I don't know. They all do similar things, Dixon. So this protein of toxo, it's called ROP16, is a key virulence determinant. ROP16.
I wonder where it comes from, Vince.
Where does it come from?
It comes from the rope trees. That's ROP, stands for rope trees. Rope trees are those intracellular organelles.
Rope tree, yeah. Tree, yeah. Exactly.
It's an odd name for an organelle.
So this dysregulates STAT6.
That's remarkable. So now he's made progress to understanding the exact underlying mechanism by which this organism survives.
You know why that is interesting besides understanding the pathogenesis? It gives you more insight into how the normal host functions. That's why viruses are useful to study. It gives you insight. They are probes for host functions.
You're absolutely right.
And so here is also...
you are absolutely right.
Parasites.
So take a guess at how many interleukins were discovered after parasitologists entered the molecular biology world.
15.
At least. Maybe
more.
Christopher
Hunter at UPenn, he works on toxo-NILs.
Right.
And in fact, I talked with him when he visited here a few months ago.
Okay.
And I recorded about a 10-minute conversation.
Cool.
On toxo-NILs. I have to dig it out.
Leishmania induces a bunch also.
Yeah. Are we going to talk about Leishmania sometime?
We will.
All right, good.
And Steve Beverly's work and all those kind of people that are still out there working.
So we have done 2 episodes on toxo.
We have.
Will that do it?
Well, we could go on and on and on. Of course, people working on toxo wish that I would do more or interview them.
You know what we could? We can do a survey of all the important parasites for the first few...
I think trypanosomes are next, actually.
Trypanosomes. That would be great.
I would like to take trypanosomes on next.
You know, we do a survey, and then we come around and start doing detailed stuff, and maybe we can talk to people.
Sure.
We'll invite them here, give them lunch.
Hey, watch them acquire these parasites before our very eyes.
I have a few emails, Dixon.
Okay.
One is from Kevin, who writes, I was Googling the internet after listening to TWiP 9, where there was a mention of the tongue parasite isopod. This is a parasite of fish. Goes in and replaces its tongue. Do you remember that, Dixon?
I do.
From the looks of the published paper, which he gives us, this is the real deal. And here is the paper, and he says, I will never eat sushi again. Paper is from the World Journal of Fish and Marine Sciences, Variation in the Morphological Features of Isopod Fish Parasites. So they actually exist.
Yeah.
Isopod parasites, what do they do? Well, they don't say here.
They're ecto.
They associate with many species of commercially important fishes.
True.
And may kill them. And here's a picture of them.
Yep.
It's large.
Yep.
What does the word isopod mean?
The same size legs.
Here it is shown on the gill area of the
Yeah, these are ectoparasites. They're mostly ectoparasites. And in fact, people who salmon fish, right, can tell how long the salmon has been in fresh water.
By?
If it doesn't have any sea lice, it's been in there for a long time.
So they come off when they go
These are called sea lice also.
Interesting.
And they come off because they can't survive in fresh water. And that's how they get rid of their parasites, Vince. And many times, that's when you see a salmon jumping out of the water for no reason whatsoever, it's shaking out the parasites from its gill rakers. It's getting rid of them. At last, they're free.
Free at last.
To acquire freshwater parasites.
Yes, yes. Hey, Dixon, you went trout fishing recently. Do they have ectoparasites?
Of course they do. They all have
Everybody does.
They've all got them.
Kevin says, also for her birthday, I got my mom a copy of the book Vincent recommended about Henrietta Lacks, and it was the best gift I've given anyone in years. Thanks.
Wow.
The Immortal Life of Henrietta Lacks. Yep. That was a TWiP pick of the week. Mitchell from North Carolina writes, Hi, Dr. Despommier. This is for you.
Hey, Mitch.
I am a microbiologist currently working to develop a TB diagnostic device, and I must first let you know that I very much love your show. I was somewhat skeptical that I would be as big a fan as I am of TWiP when your first episodes came out, but I really like the distinctly narrative style of your show. The stories and anecdotes are really great and provide a much-needed aspect to science communication. The real reason I'm writing, however, is because I got a word spy email. I get a word spy email every week about neologisms, and your name showed up in the first ever recorded citation of the word farm scraper. Got it? Farm scraper?
Got it.
It doesn't say that you coined it, but your name is now forever associated with this. Maybe this is a bigger deal to me than other people, but I think it's pretty fantastic. Here's the link.
Thank you.
Here. Words by Farm Scraper.
Uh-huh.
So, a high-rise building used for growing crops. And the earliest citation, Dr. Dixon Despommier, professor at Columbia, believes that vertical farm skyscrapers could help feed cities and fight global warming. The price tag, roughly $200 million per farm for startup costs, and each is self-sustainable.
I'd revise that down to $40 million because we're now going to build one in Newark.
Dixon, you are now the founding person for Farm Scraper. What do you think of that, Dixon?
I'm flattered, and I feel it is a big responsibility to continue the work.
Anyway, I really enjoyed the show. I also heard and enjoyed your recent interview on futures in biotech.
Great.
You read a lot of emails that say your show should be every week, but I'd like to give my controversial opinion that once or twice a month is perfect.
Thank you for alleviating my guilt about not having done this more often.
I think it gives you more time to go deeper into the history and the stories, and it allows me to focus on the show. I often don't get to listen to the whole thing at once, and I will often end up with several episodes piled up. I don't want to feel like your show comes so often that it loses its intensity. Thanks for the great podcast. Keep them coming.
We'll try.
Greg writes, you've probably seen this, but it's an interesting article concerning Toxoplasma and its possible influence on the personalities of entire nations. It's from Discover magazine. Have you seen this, Dixon?
Have not.
A nation of neurotics blame the puppet masters. Once again, I hear the siren song of Toxo, the parasite that dwells in the brains of 50 million Americans. Is that true, Dixon?
Something like that.
In the brains?
Yeah, well, remember the pseudocysts. That's why they form there. The rest of them are killed. See, I didn't actually distinguish that feature, but I will now.
So this is an article which is on a paper which has come out, which says, can the common parasite Toxoplasma influence human culture? And what he says, 3 observations. 1, toxo-infection rates vary from country to country, as we talked about.
Right.
2, psychologists have measured some of the personality traits influenced by toxo in these countries. People with toxo tend to be more self-doubting and insecure, among other things. Among the differences in men, toxo is associated with less interest in seeking novelty. Toxo-infected women are more open-hearted. 3, a nation's culture can be described, at least in part, as the aggregation of its members' personality. So is there a relationship between prevalence of toxo and its culture? And he argues, yes. He selects a few key features and he goes over them. So the French are neurotic, 45% toxo. Australians are not, 28%. That's not good study. It's not a good...
no, I think this is too subjective.
But I'll put a link in the show notes anyway for everyone to look at. Thanks for
and
that,
avoid
Greg.
that kind of data in your own work.
Yeah, because you have to control for many other factors in these studies, and they're not
Of course.
Greg, by the way, is from Virginia Beach.
Okay.
Our old friend Jim writes from Virginia. Vincent Dick, the July 2nd podcast from Science, has a 3-minute segment, 25 minutes into the podcast, that discusses the question, do parasites affect intelligence? But does not make reference to Dr. Despommier's comments made in a previous TWiP about the historic impact of hookworms in the southern U.S.
That's right. Too bad.
They should listen to TWiP.
They should. There's a well-documented study on this.
Does this current work confirm or extend Dr. Despommier's hookworm discussion? Listeners can see the transcript of the podcast, and he gives a link to that.
Dr. Peter Hotez, by the way, if I may interrupt.
Yes, you may interrupt.
Dr. Peter Hotez at George Washington University, chairman of microbiology and tropical medicine, is a world expert on hookworm, and I get most of my knowledge from his bon mot. And it was through him that I saw these studies, and he's a clinical pediatrician by training. So I don't think he would exaggerate the influence of the parasite that he knows very, very well with the patient population that he knows very, very well. So it's from his analysis of prior data sets that he concludes, and other people too, that hookworm disease alone lowers a potential intellect of a particular person. And it stays that way.
All right, let me play the 3-minute clip and you listen.
Sure.
Finally today, David Grimm, online news editor of Science Magazine, is here with a wrap-up of some of the latest science news, including a story about whether parasites lower your intelligence quotient, or IQ. Dave, how would this happen?
Well, Rob, the researchers blame it on something that they call the parasite stress hypothesis. It sounds really complicated, but essentially what it means is that when children grow up, as they fight off infections, their bodies have to expend a lot of energy to fight these infections. But when you're growing up, there's something else that requires a lot of energy to develop: your brain. It turns out your brain needs a lot of energy too. And the idea with this hypothesis is if your body's spending a lot of energy fighting off diseases, then your brain development is going to suffer, and hence your IQ is going to suffer along with it as you grow up.
First of all, though, aren't there higher incidences of parasites in poorer countries? And poverty might better explain intelligence levels?
Well, that's a good point, Robin. There's actually a lot of things that could account for decreased intelligence. When another group of researchers published a very controversial study earlier this decade, they actually tried to estimate the IQ of various countries, over 100 countries. And as you sort of alluded to, Rob, they tended to find lower IQs in a lot of these more third world countries. And so one explanation is, well, maybe it's the poverty, maybe it's the lack of education. There's been other hypotheses that maybe it's linked to nutrition. Poor nutrition, your brain's not going to develop well.
The smarter people might tend to leave?
That could be a reason too. And so what the researchers tried to do in this new study was eliminate as many of these factors as possible. They went back to this study that had been done a few years ago, and they tried to see, could they find a factor that really explained the lower IQ? And they looked at a lot of things that we just talked about, nutrition and education and other things like that. And what they found was the strongest factor that seemed to be correlated with IQ wasn't any of these. It was how much infectious disease these countries deal with. Things like malaria, other diseases caused by viruses and bacteria. It seemed like the countries that had the highest incidence of these infectious diseases also tended to have lower average IQs.
Any concerns about bias of IQ studies themselves, though? That they're geared towards people of certain cultures and not others?
Well, that's definitely one criticism of the study, is that the entire foundation of the study, which is IQ, is a very controversial topic. Some people say it has nothing to do with intelligence, and some people say, like you said, Rob, that the measure of IQ is very biased towards Western cultures or more developed countries, and that it puts people in other countries at a disadvantage. So to really start to buy into the results of this study, you first have to buy into the whole idea of IQ and that it's really an accurate representation of intelligence. But if you do buy into that, what the authors here are saying is, look, we really try to control for a lot of these other factors. And even after we do that, the strongest association we're seeing is between IQ and parasites.
So another argument for helping these countries to eliminate the high incidence of parasitic diseases.
Exactly.
Okay.
Okay, Vince.
What do you think, Dave?
I think it's an excellent start at a large problem. Allow me to expound on this a little bit, because I have my own hypothesis, and it's based on two different sets of studies. The first is, of course, the correlation between growth retardation and hookworm infection. And that was well documented during the 20s in this country, when we were busy sanitizing and creating outhouse development, let's put it this way, and to convince people to use them. And we would measure in whole communities the growth rates of children prior to and after the institution of proper sanitation. And there was a huge difference. And in fact, there was never the ability of the earlier population to catch up to the growth curve of the latter population. And they also started the studies on IQ. And there was a huge difference in the two populations. But you could argue that they were from something else too, maybe nutrition, maybe environment. All these things would change as the health. Right, that's study number one. And those are well documented, but they're early studies and they're in the 20s. And you could say, well, they were biased in terms of race and that sort of thing, which is true, by the way. But when you tease out all those factors and leave them on the side and just look at the way people develop as children, that no matter how you slice it, if they're heavily infected with hookworm, they're not only anemic, but they're also behind the curve in terms of learning, and they never catch up. All right? Let's just leave it at that. But how does that work? How could that possibly work? So that's my second hypothesis. The second hypothesis is based on some wonderful studies done in the 60s and 70s, 1960s and 1970s, with regards to lead poisoning and its effects on learning behavior. And in fact, children that are exposed to lead at an early age get behind the curve and they can never catch up, ever. And one of my colleagues, Joe Graziano, and his group in the Department of Environmental Health Sciences has studied this extensively in other countries, including Yugoslavia, now separate Croatia, etc., and in places like Bangladesh as well. And they have found unequivocal correlations between the exposure to heavy metals, particularly lead, and the inability to learn at a certain rate. Now then, how can you connect these things together? Allow me to suggest the following. Hookworm. In fact, I think I addressed this issue a little bit on Radiolab, but hookworm sucks blood and derives the oxygen from the blood and creates anemia as the result in heavy infections. And it eats your villus tissue, which continues to bleed after the worm leaves because it uses anticoagulants to make sure that the blood doesn't clot up so it can actually extract the oxygen from your blood. The point is that in anemic situations, the body has some recovery schemes. All right? One of the schemes is to recover all the iron. Now, how does it do that? Once the red cells break down in the gut tract that the parasite doesn't use, by the way, the parasite just takes the oxygen out and throws away the whole red cell, and in fact, all of it. Maybe it saves the plasma for its own purposes, but the rest of it is thrown away. What happens to the iron and hemoglobin? And the answer is that there's an uptake of iron that's increased because the anemia itself upregulates a protein, which is a divalent cation transporter molecule. A divalent cation transporter molecule. So it will transport any cation, including lead. So let's assume that you're infected with hookworm as a small child, and you're living in a depressed area that's been heavily impacted by pollution from industrialization. So you've got an overlap of exposure to heavy metals and heavy infection with hookworm. This is very, very common, very, very common throughout the world. You now have the potential for hookworm inducing the anemia part and the upregulation of the uptake divalent cation molecule on the surface of the gut, not only taking back all the iron, but also the contaminants in the diets of these people, namely lead, vanadium, cadmium, zinc, any of those compounds. Now, so how does that work? How does lead inhibit your ability to learn? We know. All right? Now, Vince, you and I have sat here for how many times now? We're up to almost 100 episodes of TWiP, which I love, by the way, because I'm the innocent bystander, acquiring knowledge through Vince's genius.
No.
No, it's true. Don't deny this, Vince. You're a brilliant virologist.
I'm going to cut this out.
I don't care. But you are. And you've already been recognized for that, so don't be modest in that sense, because you've occupied a chair based on your work with poliovirus. You and I learn from each other. Let's just leave it at that. You and I learn a great deal. But how do you learn? What is the process of learning? Well, Eric Kandel, who has won a Nobel Prize for his ability to dissect out the learning process of an aplesia, found the following. An unlearned aplesia has neurological connections, which are, and I'm going to do this with both of my hands and put my fingers together so that they match mirror-like. Before learning, they have this configuration of connection. As you begin to learn, the old connections are released to make room for the new knowledge. And so your neurons rearrange themselves.
Dixon is shuffling fingers now.
I'm trying to show you how your neurons behave the moment you acquire new knowledge. They detach and reattach and make new connections, and they begin their process of incorporating the new knowledge into their base. whatever that means. Okay, for aplesia, it was a simple learning process from A to B or something like this, or red to yellow or something. But with us, it's much more complicated. So we don't really fully understand how the brain works. Remember, I said that during the Toxoplasma lecture, or I should say presentation. I don't mean to be these things to be lectures, but they always form back to these lectures that I gave, right? So, but the point is that what if something prevented the neurons from coming apart at the same rate that non-affected neurons would come apart?
Like lead, Dixon?
Like lead, Vince.
Yeah.
In fact, it's already been shown that that's exactly the mechanism that is at work, which accounts for the lead-exposed children. Now, in animal models, of course, you'd have to do this, but it prevents the neurons from detaching and attaching rapidly. The interdiction of a divalent cation forms a bridge between these two connectings.
Yeah, you could do that in aplesia, right?
You can do it in aplesia, you can do it in anything. You can slow down the learning process. You can ablate the learning process by heavy metals. Okay, so if hookworm induces the anemia and lead is the cause of the learning deficit, hey, that's A plus B equals C.
It's a good mechanism.
That's like a quadratic equation thing, all right? So now I've been touting this thing for, I don't know how long, but I hope some people out there are listening that know more about this than I do that can add some fuel to the fire and say, no, it doesn't work that way, so it's this way. But
it's a specific mechanism where you might say, well, people who get malaria every year, they're just physically unable to learn and work, but there's no specific mechanism. This is specific.
This is very specific. So it's testable. You can actually, you can make maps.
Yeah.
Right? You can go into an area and you can survey for hookworm first. And you have those maps. And I think Peter Hotez has already done a lot of this work. And then you could go back and you can test the environment for heavy metal contamination. And you can overlap these. And then you can do your IQ testing in groups that are not and are. And now the groups that are exposed to the heavy metals are probably, by the way, malaria induces anemia too.
Okay.
Yeah.
Could be similar stuff.
There's a lot of things that induce anemia. And once the anemia is induced, the heavy metal transporters are upregulated because they want to conserve all that iron no matter what.
Yeah, no, it makes perfect sense,
So
Dixon.
that's my take on all of this.
So, Dixon, for closing comment, parasites have a huge impact on world development.
Enormous.
We should get rid of them.
Well, some of them can be gotten rid of by just farming in a different way because we use human feces as fertilizer, and that spreads the parasites.
Right.
That's the basis for this other project that I'm involved with.
Sometimes a very simple thing. For example, in Bangladesh, the people collect date palm sap in big vats, and at night, do you know who goes to feed in them?
Sounds like the bats might do that.
And they defecate and urinate, and they put viruses in, and then the people drink the sap, and they get viruses.
Oh, my gosh.
So a simple solution is simply to put a lid on it.
Put a lid on it.
You don't need antivirals. You don't need vaccines.
No.
A lid.
Just a behavioral change.
Sometimes it's all you need, right?
A behavioral change.
Dixon, it's been a pleasure talking with you again. Good to see you. I missed you.
Same here.
Don't be scarce.
No.
All right.
Try not.
Hope we can do it again soon.
Yep, me too. You're off to Bozeman, Montana.
I'm off on Saturday to Bozeman to do TWiP.
And I wish I was joining you, but I'm
live.
not.
I'm sorry you can't. You would have had a good time. And you could have taken me fishing.
I could have, but I will do that anyway.
You do it another time.
Yeah.
All right. If you like TWiP, subscribe in iTunes. It's free. You get the new podcasts as soon as we upload them. And if you like it, make a review in iTunes. It helps us stay on the front page of the medicine podcast section and more people see us. You can listen at microbeworld.org/TWiP, and we have show notes there as well. TWiP is part of sciencepodcasters.org and promednetwork.com, which are other websites where you can find science content. As always, send us your comments and questions. TWiP@twiv.tv.
It's a tongue twister, Vince.
You think?
I do. Say that fast 3 times.
I don't think so. Too much lead.
Right.
You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.